Okay, so is there a way that we can automate this identification process, right? So I don't know who has a background in machine learning or not. The next few slides are really very basic machine learning on what is clustering and what's classification. So if you really know this, just stay idle for a minute. We'll get back to single cell quickly. So if we talk about identifying cell populations, we kind of want to define these boundaries between the cell types, right? What is really differentiating between B cells and myeloid cells? When we do this with clustering, this is an unsupervised learning, so we don't try to learn anything from the data. We're basically trying to discover what is the structure of my data, how many groups are in my data, how far are these groups from each other, or how similar they are to each other. So we kind of try to define this boundary that if these are the cells that I call B cells, these are the myeloid cells, and there is a boundary somewhere here. Yeah. If we move to classification, this is supervised learning. So here, indeed, we, instead of just finding this boundary that differentiates the cells, we actually want to describe the boundary. I want to get a function that tells me where is this line, so that if I get now a new cell and I place it here, for instance, then I know its relation to the boundary, and then I can make a decision whether it lies to the left of the boundary or does it lie to the right of the boundary. Yeah. We do require prior information, of course. We need some labels. So I need to learn this relation from data that I have seen before. Yeah. I hope this is a bit clear. So to illustrate how this works in classifiers, so you have your data, so you have cells, again, you have measured features. These are genes in our case. You have some labels, so it tells you, okay, the first two cells are T cells, these are B cells. I will use these dimensionality reductions to describe the data, right? But you have already probably looked at dimensionality reduction, so this can be PCA, TSNE, UMAP, whatever. So if you represent your data, you hope that these T cells are closer to each other than they are to the B cells. And then your goal is to find this boundary, right? So we learn this boundary that tells us, okay, where is the blue group, the T cells, where are the orange cells, the B cells. And now if I get a new unknown cell, I don't know what the label of the cell is, I can project it to this space, right? So then it lies over here. And because I know the boundary, I have learned the function that describes this boundary, then I can say, okay, this is definitely a T cell. Yeah. Okay. So how do we do this, right? How do we train these classifiers? So let's do a little bit of equations here, because I think it helps. It's a lot easier to understand it this way. So if we have cells, each cell is described by its gene expression profile. We call it X here. So XJ is the expression profile of cell J. Then you also have another label that's YJ that tells you whether it's a T cell or a B cell, right? And this, you can just give it a number because it's easier. A classifier is simply a function, right? So it's a function W, no matter what this function is, it's just a function W. You give it the expression profile of the cell, it gives you a label, right? Just as we did here. We give it this vector over here, and it gives us a label back. That's all what this function needs to do. When we train this function, when we try to find this function, our goal is to minimize the error. So if we have data that is labeled, I already know that these are B cells and these are T cells. I want a function that gives me the minimum error in such a way that it does not predict a B cell to be a T cell or a T cell to be a B cell.